So when we talk about cells, again, I'll have to write with my finger until I get the pencil recharged. Cells have membranes, and we've talked about membranes a lot. We know that cell membranes define the cell, right? We know they're a phospholipid bilayer. These are things you should know. They should be already in your notes, so I'm not writing them down. But do you know that they should be phospholipid bilayers? You know the different kinds of proteins that should be in there. And we've talked enough about that you should be aware that that's important. And we've said something about the idea of there being some kind of way of telling what gets into a cell and what doesn't get, and what gets out of a cell. We've also talked about this thing called, we mentioned, not talked about, we're going to talk about it today a little bit. We've mentioned this area, this space on the outside of the cell membrane. It's outside the cell membrane. It has a name. I'll give you the name in a second. And this stuff, it's made of proteins, and they're attached to the cell membrane. They can be tethered. The proteins can be anchored. There can be all kinds of ways they're attached. But there are these things called glyco, meaning sugar, right? Lipids. So maybe these are lipids that are tethered to sugars. There are these things called glycoproteins. Glycoproteins, again, are sugars that are attached to what?
Protein.
To proteins. And these both act as signals, almost like a key in a lock or a flag. So why would anybody put a flag out? Why would someone, there's a war going on, they're shooting back and forth. Why would you want to have a flag? To claim an area, I like that. What else? What's another reason you'd have a flag? To tell which territory is yours, I like that. So you're demarcating that something belongs to you, right? So another way these can be used is as flags. So again, what do we do with flags? We mark territories. We mark territories. This is ours, right? We mark what is ours. We mark what's ours with a flag. So when we talk about glycoproteins and glycolipids, they could be involved in signaling. In signaling what? In signaling what belongs to us. Someone close the door, please. What belongs to us? It could be a lock and key. It could be an enzyme in that space outside the cell wall, outside the cell membrane. What belongs to us? And one example of what belongs to us is this idea of blood groups. You've heard of blood groups before?
Yep.
What blood groups have you heard of? What blood groups have you heard of? For example, of what belongs to us. What blood groups? A, B, O, AB. You've heard of these, yes? And then there's plus or minus. It's RH, RH plus or RH minus. There's actually thousands more. There's thousands. I'm going to say thousands. It might not be thousands, but let's not say thousands, say many more. Many more markers, we call them markers, that tell us, they act like flags telling us this belongs to us. So if you're going to get some kind of transplant, you need to have some very similar markers. And so to get a transplant, they have to test all these different markers that are on the outside of all your cells. Because if they're different, your cell will say, this doesn't belong to us. We got to kill it. There's so many markers that no two people have the same markers. They don't exist unless they're twins. Unless you're twins, unless you have a clone. A twin is a clone. An identical twin is a clone. And the only way you're going to get the same markers on the outside of every cell is if you have a twin. So unless you have a twin, you have no donor that's perfect. So no matter what, we have to when you get a transplant, you have to take medicines that calm down your immune system. They kind of make your immune system drunk, if you will. So they can't see everything clearly. It's a little fuzzy. If we can do that, then we can transplant. But even then, you have to get it close. So we can't turn your immune system off completely, if that makes sense, right? Because then bacteria would kill you, correct? So viruses and bacteria are going to kill you if we turn off the immune system completely. So we got to do something about that, and the only way we can do anything about that is just to make your immune system act a little funny. Just calm it down, tune it down. So these markers, the way they work is with these glycolipids. Imagine taking a lipid. Remember, it's a carbon-hydrogen chain. The carbon being delineated by them, and then and at the end of that, you add a lipid, a carbohydrate. To that lipid, you add a carbohydrate. So you do something like this. Let me make it smaller. So you do something like this. You add a bunch of carbohydrates. They don't all have to be the same carbohydrate. Maybe the carbohydrates are in a ring, or maybe they're in a double ring, or maybe they're in a chain. There's all kinds of things that could happen to this. What's really cool about these these glycolipids and glycoproteins is they can take on a bunch of more different shapes. And then each of these ends up being a different flag. That's how you can have 1 billion people, more than 7, 8, 9 billion people, and everybody have a different set of markers. Because each protein, each lipid that sits on the outside of that space can have a different shape. And that's because, you could add all kinds of carbohydrates and lipids on the outside. There's so many different combinations. This purple space on the outside of here is known as the extracellular matrix. Why is it extracellular? Why extracellular? Why is it extra? Say what now? Very good. Because it's on the outside of the cell membrane. So the cell membrane's here. Everything inside the cell membrane is part of the cell. This is outside the cell, so it's extra. It's an extracellular matrix. It's attached to the membrane, but it's on the outside. And you'll see drawings with all kinds of crazy things in here. Think of it almost like a jungle out here of different molecules that stick out. Some are receptors, so they act as some act as enzymes. Some act, the lock and key idea that's happening in the extracellular matrix. Some act as signals. Some are being, like I said, receptors, or maybe they're markers, flags. But all that's happening on the outside of the cell. We haven't even gotten into the cell yet. Now, inside the cell, we're gonna divide this into two different kinds now. We got eukaryote. Let's talk about eukaryotes. And yes, eukaryotes are spelled with a K or a C. One's British and one's an English spelling, American English spelling. I don't care which you spell it, but it should be something like this, eukaryotes. Eukaryotes, all cells, and I should mention this, all cells have a cell membrane. All cells have the extracellular matrix. Should I draw that? Why not? I think I can handle that. All cells have this extracellular matrix that act as a flag of some kind. Hey, this is one of us. Some of them have like a mucus layer, but there's this outside that's... Now, eukaryotes have something special, and this is going to be key if you're going to understand, and you're going to have to be able to identify the difference. Eukaryotes have membranes inside the plasma membrane. They have membranes inside the plasma membrane. Does that make sense? They have membranes inside the cell. Does that make sense or not? Yes? All right, so they have spaces. They have compartments. Remember that from the video, compartmentalization. They have many compartments inside. How do we define a compartment? How do we define a space in a cell? We use what? Say what? Yeah, how do we make a room in a cell? What do we use? What do we use to separate the spaces to call it a room? To call it a room, you have to have something separating it from everything else, right? Like this is a room, and there are these things called room walls, right, to separate us from the rest of the building. If you're going to have a cell, you have to have something that's going to separate this big space from any other space that you create in here. What would you call that? What's the barrier? What is the barrier? What barriers do we use? You're right. You're absolutely right. But I'll give you a clue. I already wrote it down. Membranes. That's right. We use membranes. We use membranes to... I really can't stand the way my finger writes on this. Let's see if my pencil is it charged enough for me to be able to... So, membranes. Can't you take a piece of paper and like unwrap the whole thing and put it down and then eat it? That way we don't have to like listen to the crackling every 5 seconds. She's so nice. She's trying to be real quiet about it, but every time she crinkles something, he's like, crinkle, 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 crinkle. And then it's chocolate, so there's like chocolate all over her fingers. It's all over her pencils. She's licking her pencil. It's just a mess. It's cute to watch, though. All right, so membranes on the inside of the plasma membrane. So what's key is that this eukaryote has many rooms. One of the rooms that make it very distinctive is this thing. And what do you think that is? That's right. That's called the nucleus. That's called the nucleus. It is made of a membrane, creating compartments. Inside this compartment, inside the nucleus, you have DNA. In eukaryotes, it's linear. It's linear DNA. It has 2 ends, just like we talked about. There's another room inside the nucleus. There's a whole other room here that's called the nucleolus. Very similar. So there's 2 rooms Now, I'm going to do one of these things if you don't mind. I hope you don't mind, but if you do, it's too bad. It's going to look something like this. Have you seen these things before? They do it in books a lot. Have you seen that in books or not? What am I doing? Why am I doing this? That's right. I love that. Zooming in is correct. Yeah. Brilliant children. That's right. Zooming in. And when we can zoom in, what we see if I zoomed in, and let's say I made the nucleus, I'm going to make it purple. All right. Actually, maybe I shouldn't because I'll make it yellow. So I'm going to make the membrane yellow. This inner membrane yellow. The white one is the plasma membrane or cell membrane, right? Everybody get that? That that's a cell membrane? Yes, you'll have to identify what each is and what each does and the difference between it and the others I'll show you, yes. If we look at this one, which is called the nuclear envelope or nuclear membrane, then what we'd see is this membrane. It'll be made of lipids. Is there someone knocking on our door?
Yes, sir.
Who's knocking on our door? What's going on? Is it not time to leave yet, is it? Oh, someone went to the restroom. There are holes in the membrane. There are these holes. These holes are called nuclear pores. They allow things in and out of the membrane. You know what? If you don't mind, I'm going to undo that, and I'm going to draw it over here. And nuclear pores they allow, bless you, stuff in and out of the nucleus. Now, DNA can't leave. So DNA cannot leave the nucleus. Is that clear? DNA cannot leave, but RNA can. That's going to be very important. This is one of those things that foreshadows. You've read books before, I'm assuming, and you know these things. This thing called foreshadowing, where they tell you something later in the story comes back, and that's the bad guy, or that's the good guy, or that's the thing that was a secret to everything. Well, this is one of those foreshadowing things. DNA can't leave the nucleus, but RNA can, and these pores let things in and out of the nucleus. There's another organelle in this eukaryote that's called, it looks like this. Let me see, color coded. I'll make it blue. There's a lot of these, and again, they have a membrane. They're folded. There's two membranes. There's double membrane. So this one has a double membrane. And it's called a mitochondria. And mitochondria have double membranes, mitochondria process energy, they make ATP. We'll talk about what ATP is later. So you should know that's why they call it the powerhouse of the cell. You've heard of that before, I'm sure, in elementary school. They make process energy, the double membranes, and they have their own DNA. That's really interesting. They have their own DNA. So mitochondria are really, really interesting little things. And they're very much like prokaryotes. Are they prokaryotes? No. But are they like prokaryotes? Yes. And they're inside a eukaryote. Isn't that interesting? There are these things that are organelles that have double membranes, their own DNA, and they act like prokaryotes in very many ways, very similar to prokaryotes that live inside every eukaryotic organism. Yes? Question?
Can they be mistaken as prokaryotes?
That's a good question. I suppose if they were floating around somewhere, somebody might consider them prokaryotes, but they're so distinctive. They can't exist outside the cell. So it's a good question. They're very similar to prokaryotes in many ways, these organelles. Now, that's one type of eukaryote. There's another, let's draw another type of eukaryote. Let me see if I can do this. I don't know if it's gonna, I'm gonna hate doing this, but I'm gonna go ahead and duplicate it. Now, there's another type of prokaryote that's very similar. Everything's the same. I'm gonna go ahead and get rid of a lot of this though, so I can draw some of the other stuff. So a lot of this stuff is still there. Is that clear? I'm not getting rid of that. The cell membrane's still there. I'm actually going to go ahead and leave the lines. It is a eukaryote, though. It just has something else, some other things that the other eukaryote doesn't have. Spikes. It's too thick. Oh, I'm sorry. I know I'm picky, but it just looks stupid. I'll just rewrite the whole thing. How's that, jerk? It's still a eukaryote, but it has this really interesting set of features. And the first feature I'm going to go ahead and draw out is outside the cell matrix, it has this thing called, and it's not really to scale, so I apologize for that, but this thing called a cell wall. It's made of cellulose, so it's actually made of sugar, right? It's a polymer of sugar. We learned that. It's made of cellulose. And inside the cell, this eukaryote has something else that's weird. It has, let's see what color should I make this? These things called, I'm going to go ahead and they look weird. They have stacks of membranes. They're stacks of membranes. Again, double membrane. Let's go ahead and draw that out here. They have double membranes. They have their own DNA. They also process sugar and process energy. But they make sugar. They do something called photosynthesis. And they're called chloroplasts. And they're green. That's what makes plants green. And then there's another organelle that's really weird. That's not in the other eukaryote. It's really big. It's actually the biggest organelle in the cell. It takes up a good portion of the cell, of this type of cell. These are not in this, so let me erase that. I'm going to leave the chloroplast there because I already drew the line. So I'm going to erase these. These aren't inside. The chloroplast is not inside it. Is that clear? It's above it, if that makes any sense. Does that make sense? Yes? Are we clear on that?
Yes.
Yes? People in the back?
So
Do you understand what I'm
outside?
saying? So, no. They're still all inside the cell, but this is three-dimensional. So this is further back, right? So this organelle would be further back. This is closer to you, and this one's further away from you. Does that make sense? All right. I hope it does. I hope you don't confuse it. This thing holds a lot of stuff, and we'll talk about what it holds later. But it really has a lot to do with water. And this thing's called the central vacuole. Central vacuole. So as a central vacuole, has chloroplast, has a cell wall, but it still has a nucleus and all the rest of the organelles. There's a lot of organelles I still have to cover, but this is what we have so far. The central vacuole has to do with water regulation. That's the main function. It regulates osmosis, osmoregulation. And that's not a Q, that's a G. So that's water regulation. Are we clear? That's what the central vacuole is doing. So these two things, let's go ahead and look at them. They're both eukaryotes. This one, they both have mitochondria, correct? They both have cell membranes. And honestly, someone said in the other class they should do a Venn diagram. She was joking, but then it turned out I kind of agree with her. I think she said it because she was sick of Venn diagrams. But mitochondria, they both have nuclei, correct? They both have nucleolus. Can't you guys just leave the door open when you go to the bathroom? They have, huh? Yeah, leave it open. Yeah. Is it me or is it cold in here? It's either cold or hot, man. I can't get it to stay in just normal. So we know that these are all the things they share, correct? They both have an extracellular matrix. Again, they both share other organelles. We're not there yet. But then there's some stuff that's different. And now the stuff that's different, that's all the stuff that's the same. The stuff that's different is what? This one has, I should tell you. These cells, these types of cells over here, they can have a flagella, or they can have cilia. Cilia or flagella. Someone check the spelling of flagella. See if there's two L's in it. One of those things I always mess up. So flagella and cilia, they're for moving. Flagella you've seen on a sperm, right? They kind of have tails. They look like this is well done. All right, good. And cilia, they're like little oars. In fact, I think Nolanix found ciliate the other day, was zooming around in the pond water. They zoom around, they eat bacteria. But you have cilia too. You have cilia in your throat. That's what feels like it's tickling. It's in your trachea and in your bronchioles. in your airways, you have these little cilia that help get the mucus out. When you cough up that mucus, that's the cilia moving all that mucus up with trapped dirt and bacteria out of there. So cilia and I should add that. So one of the things that's different is that, so these are the same, is that these guys have cilia and or flagella. All right. But this other kind has cell wall. This has chloroplast. This has central vacuole. What is this kind of thing here versus this kind of eukaryote? This eukaryote versus this eukaryote. Anybody know? No guesses? This would be known as a plant cell. And this one over here would be known as an animal cell. So one list of eukaryote, one type of eukaryote is an animal cell, another type of eukaryote. Both plants and animals have nuclei. They have these compartments inside the big compartment called the cell. So let's go ahead and talk about all the different kinds of eukaryotes and prokaryotes. Let's organize these into groups. So, because we're looking at a lot of cells, and Thursday you'll be dealing with a lot more cells. You'll actually have a formal lab write-up you're going to have to do on what you've discovered. So prokaryote can be either a bacteria or it could be an archaebacteria. Slightly different, but what's interesting about these guys is they have no internal membranes. They only have the plasma membrane, and they have round DNA, which is interesting to me. Their DNA is circular. They have smaller plasmid DNAs. Ribosomes, they have ribosomes, but ribosomes don't have membranes. So no internal membranes. So what does that mean? No compartments. No specialized compartments. Now in eukaryotes, we already talked about that. You have the nucleus, the nucleolus, you have the chloroplast and the mitochondria, and there's others we'll talk about a lot more. But here, there are no compartments, no membranes. Everything that happens in this one space. It's still a cell because it does have a cell membrane. And interestingly, it has a different kind of cell wall. It does have a cell wall, but it's different. All right, whatever. It does have a cell wall. It's just different from a eukaryotic cell wall. I shouldn't draw it that way. It's made of something different. A plant cell wall is made of cellulose. This is not made of cellulose. But it does have a cell wall. Has a cell wall, has a plasma membrane, has a nucleoid region. We call this region a nucleoid region. So there is DNA. It does have DNA. All living things have DNA. It has a plasma membrane. It is a cell, but they don't have all the fancy stuff that eukaryotes have. Are we clear? So the prokaryotes are very simple. And I believe your book calls the difference between this and a shack. A house and a shack, right? Or it's not the book, maybe it was the MIT lecture, but in any case. So if you think of a prokaryote, you can think of a prokaryote like a tent or a shack, right? One room shack. You got your bathroom, your kitchen, your bedroom, all in one space. There's no, your toilet's right next to the bed, it's right next to the stove, right? That's how people used to live. You realize that? Okay. Usually they put a toilet outside, but when they had indoor plumbing, the toilet would be right there. There's no room, no walls, nothing. It's just all in one space. As if we all lived in here. We had a kitchen there and a bathroom there, but there's no rooms. There's no separation. So but in a eukaryote, it's more like a modern house where you have rooms for everything, okay? Eukaryotes tend to have rooms for everything. And there's more than one type of eukaryote, just like there's more than one type of prokaryote. Eukaryotes, we know about animal. But we have animals. Animals have a really, really close cousin, which a lot of people don't think are close cousins. In fact, we used to think they were plants, but they're very close to us in how they do things. We call them fungi. So mushrooms are very close to humans. It's interesting. Their DNA, they have similar genes, they do similar things. They don't do photosynthesis. Obviously, they eat dead things, right? That's what mushrooms do. They're necrophores. They break down material, organic material. So they are obviously very different. But if you compare fungi, the mushroom to us, and then you compare plants to us, mushrooms are much closer. And then, of course, you have plants, and you have these things called protists. Now, that thing that you saw swimming around in the pond water on Friday, that was a protist. Protists are interesting. They're kind of in between a lot of these different groups, right? They can be a lot of different things. That you can have an amoeba that changes shape. It could look something like that. You can have a ciliate that looks like this. All right. You can have all kinds of different things. The key is that they all have what? They all have a nucleus. They all have organelles. They have all the other things that you'd expect to see in a eukaryote, but they're single cell. Protists tend to be single cell. Some are multicellular because they're so different. It's a big group, mostly. Thank you. Plants obviously are different from animals. I don't know why you put anything away. I'm not done yet. Let me finish my sentence. Rude. You should be writing it down. So, protists tend to be single cell. Plants obviously are a big group, and they do all kinds of things: trees, grasses, etc. Fungi, as you know, are like mushrooms, but they're also molds. You know, the stuff, that slime that grows in your bathroom is also a fungi. It just doesn't have a fruiting body. It doesn't produce a mushroom. That's all. And of course, animals come in all different sizes, including centipedes, by the way. Where's the what? What slimy stuff? If you've ever seen mold grow in a bathroom, you know, it's like a pink, or it could be All different kind. Yeah, that you've never seen like a slimy coating in the bathroom at all. Any bathroom you've never been. Okay, well there can be slimy stuff in the bathroom. It's called mold. It's common.
I've seen mold before, not bathroom though.
Okay, well bathrooms are a perfect place for mold because it's very wet and often dark. So mushrooms need wet and dark.
I got an automatic light, so it's safe.
Well, whatever. The point is, mold grows in bathrooms and in basements and underground. All right, take it easy. What are plants? They're green. There are eukaryotes. What? Chloroplasts are some of the characteristics that make plants. Most plants have some type of leaf, but they again come in all kinds of varieties. There are leaves that come in all kinds of shapes and sizes. There are leaves that come off stems like this. There are leaves that are more like needles, like pine needles. Do you know pine needles are leaves? Did you know that?
Yeah.
So they're actually leaves. They're leaves that are evolved to deal with the cold. There are plants, most plants do photosynthesis, but there are plants that are parasites. They don't really do photosynthesis per se. They actually steal the sugar from other plants. There's some ivies and things that have, there are obviously some that do a little both. There are plants, there are underwater plants or water plants. There are desert plants.
What's the plant that takes sugar from other plants?
It's a parasite. I'll have to look up what it is, but there's more than one. There are plants that produce fruit. There are plants that produce nuts. There are plants that produce poisons. There are flowering plants. There are plants that don't flower. There are moss. There are plants that there's a lot of different kinds of plants. So there's a lot of variety here. They're all eukaryotes, though. All of them have a nuclei, and they all have chloroplasts inside them, or at least say what now? No, it's all right. What were you saying?
What about the ones that move and bite?
They don't move.
The fly traps.
The Venus flytrap, they can't get up and move. I should be clear about that. They do move. There are plants that move, but they just don't get up and move anywhere. So the way they tend to move is they tend to swell some cells open. So if you think about it like in Minecraft, if you have a bunch of cells that are laid up, remember they all have cell walls, so they can keep a really rigid shape. And what happens if you have these cells like this that make up a stem, and then these guys over here, these swell up? What happens when these swell up and they get bigger? Well, it's gonna turn the stem. The stem will turn this way. You see, and that's how sunflowers. That's how they follow the sun. Sunflowers tend to follow the sun as the sun moves across the sky, and the way sunflowers do that is they swell up certain parts of their stem, and so they turn and they follow the sun across the sky. Venus flytraps also can close and trap bugs. They do that to get nitrogen because they live in poor night. They don't eat the bug for energy. They don't need it for energy. They do photosynthesis. But they need the bug to get nitrogen because they have to make amino acids. Plants make amino acids because they are living creatures. They use the same biomolecules we use. In fact, that's why we eat them. Does that make sense? All right, next.
What you wrote under protis, what's that?
Single-celled mostly. They're mostly single-celled. There are plenty of, I say mostly, I can't really give you a number. I would say that there's a lot that are multicellular, and there's a lot that aren't.
What's the drawing?
This is a ciliate. It's an organism, a type of organism that has cilia. Remember, we said that one of the things that animals can do, and these are not animals, by the way. These, again, are not animals. But one of the things that protists and animals can do is move. So they have these things called cilia. There are some that have flagella. And the flagella allow them to move. Those are types of movement. This is an amoeba.
So they do have flagella.
Like Osmosis Jones, if you watch that cartoon, is an amoeba. Or it's an amoeboid cell. It's actually supposed to be a white blood cell. But in any case, it changes its cytoskeleton. It can move and squeeze through spaces. And it can actually solve puzzles. It's a really interesting organism. So ciliates and amoebas and organisms with flagella can all move very quickly through water.
Can you rewrite the word?
Just write it any which way and spell it at home. Amoeba. But I'm moving on. We're running out of time. So fungi, the fungus that you're kind of used to seeing in storybooks, looks something like this, right? It has like some mushrooms. Sometimes they'll put it's red, and they'll put white spots on it. So mushrooms are fungi, but and by the way, mushrooms are just the fruiting body. Most of the fungus is underground. There are mushrooms that take up whole forests, that one organism is underneath the entire forest, and then all you see is this mushroom in the field. When you see a mushroom, most of the organism is underground. There are...
Sprouts up from under, like you got so big that's and then like sprouting up.
Yeah, these are full of their babies, and the babies are in little things called spores. Focusing. Spores get that when the fruiting body gets ready to explode, it shoots out its spores into the air. You breathe in these baby mushroom all the time.
How do you spell it?
Spores. Spores. This is a mushroom, yeah. Or it's a bad drawing of one.
Wait, can you spell back up? That was too fast.
No, I can't. So we have to get moving. So spores. Again, anything that you miss, just look it up on the video. I can't say it any other way. All right, then another form of fungi is mildew.
That's fungi?
Yeah, that's a fungus. Athlete's foot. It's a disease. It's a disease called athlete's foot. The fungus gets underneath the bed of the nails and in between the toes and starts an infection. But it's...
He told us that, like, from getting athlete's foot, you pee on your feet.
I don't know about that. Let's just move on.
I've heard that.
Yeah, there's a lot of things I hear. I don't know how much of it's true. Let's just move. Let's just be clear. Let's stay with the facts in this class, if that's okay. Mildew, athlete's foot. These are examples of fungi. Yeast. From yeast infections to making bread and alcohol.
So we're eating fungi?
You are eating fungi. Or the cooked remnants of the yeast is what you're eating. bread is wheat flour that's mixed with other ingredients, and then you mix yeast in, and the yeast eat that bread, that wheat, and they produce a gas called carbon dioxide, which makes little air pockets in the bread. That's what the rising is. And then when you bake it, you kill the yeast, but you also cook the bread, and then you eat bread. That's what bread is. Now, animals, obviously, you know what they are. They are anywhere from human to things like insects, to things like, other mammals, birds, fish. The list goes on, right? So there's a lot of animals that I'm sure you're aware of. The fungi can be multicellular. As you see, the mushroom is obviously multicellular. Or it can be single-celled, like a yeast. A yeast is single-celled. Proteins can be single-celled or multicellular depending on their phase of life. They could start single-celled and end up having a phase of their life where they're multicellular. Plants are multicellular. Animals are multicellular.
Can you
So,
ever go from multicellular to single cell?
yeah, the protists and fungi do, not humans, right? We do start off as a single cell. When your mom and your dad got together and made you were one single cell. And then you started to do a process called cell division, do the cell cycle, and do something called mitosis, and you became you. You made it a trillion cells. You went from 1 to a trillion cells. So, yes, you can go from 1 to multicellular. So depending on the life cycle. So life is very complicated, and cells can be very complicated. And if I were you, I would watch and take notes on the video I posted yesterday. That's not the class video that I posted that was for fun. I'm going to assign it eventually. So if you have time, I would watch that. It's a very interesting video. I think you'll have fun with it. So let's redraw what we drew yesterday and add to it. We drew a cell. We drew a nucleus. There's a nucleolus. We said there was this little area on the outside. I'll say this again for those of you that were confused today. There's an area on the outside of the cell that is called the extracellular matrix. It's this region out here. It's made of all kinds of proteins, and yes, you could have drawn a bunch of crisscrossing proteins starting in the membrane. They're with all kinds of different functions. That is a matrix. That is correct. But this is the extracellular matrix, right? This is outside the body. This extracellular is connected to the intercellular matrix that's called the cytoskeleton. So there's a cytoskeleton that connects, that goes actually from the centrioles, it radiates out and branches and forms.
What's that?
I don't know what's going on over there, but I'm assuming.
I think you misheard me. I asked if you can go from multicellular to single cell.
Yes.
So you can go from single cell to multi and multi to single.
Yeah, both. You were one single cell, and you turned into a multicellular. So yes. And can you go from multicellular to single cell? Your father is a multicellular organism. He produced a single cell that became you. Well, a single cell that combined with another single cell that your mother made to become you.
If you die and your body decompose, can you be a single cell?
No. Once you're dead, you're dead. It's only fantasy that has you coming back as zombies and such.
How long afterwards do your cell live?
Not long. They die pretty quickly. All right, so let's be clear about this. This is what I hope none of you put as the answer to today's answer of extracellular matrix. This is the cytoskeleton. Everybody should know what that is. It's the cells are not just bags of.
Wait, the white is the membrane?
Yeah, I labeled it, didn't I? I can't tell if you can tell colors.
I was just making sure.
Okay, I don't know if you can tell colors. Can you tell the colors on the screen?
Yeah, but I was just clarifying.
I'm just saying, can you tell that this is white and this is gray?
Yeah.
Okay. So this gray is the cytoskeleton. It is inside the cell. Skeletons are inside the cell. And the blue is your nucleus. And then you have your nucleolus. And of course, we drew yesterday the mitochondrias. You can make it a different color if you'd like. Let's make it red. But there are other membranes in this cell, and one of the other membranes that we have in this cell, and this is a very complex structure. It's not as simple as you might think. There are these membranes that go back and forth. And because this gets so complicated, I'm going to erase this whole section of cytoskeleton. You have to understand the cytoskeleton is still there. Don't think it's gone. But this is a three-dimensional structure, and I only have 2 dimensions to work with, so I have to continuously erase and draw different structures.
I'm confused.
Yeah.
The gray. The
Cytoskeleton, yeah.
I don't see which one that is.
Oh, I thought that was white.
It's the what?
It's the gray. Do you see this? Do you see this? These lines? Do you see these lines?
Yeah,
This is the cytoskeleton.
but it's white.
I asked you if you could tell that this was gray. You said yeah.
Because I didn't know you was about to write the skeleton.
Huh?
Before you wrote
All right. I don't want to argue with you. Can you tell that this is gray? Yes or no?
No.
All right. Can you tell that that's not white? Yes or no?
Yeah.
Okay. That's the cytoskeleton. Carly, if you no offense, if you got a giggle, go in another room, go in the hallway. So you got this gray. That's your cytoskeleton. It gives itself form. It acts as roads inside the cell for things to get moved around. So that's the cytoskeleton in the cell.
You said, what's the functions of the cytoskeleton?
I have to get going, but it gives itself form. So one really big and important organelle is a membrane. It's actually, it looks like I'm drawing this tube, but it's actually more of a bunch of sheets of membranes, if that makes any sense. I don't know if that word makes sense to you, but sheets of membrane that sit in the cell.
What's the name again?
I'll tell you the name of it in a second. But this is called the endoplasmic reticulum. And it has two parts. It has a rough and a smooth. The rough has these little ribosomes on it. And the smooth obviously does not. So the short word for this is RER for that's rough endoplasmic reticulum.
The rough part has what on it?
Ribosomes. It's another organelle we'll talk about next, actually. And then there's a smooth ER, which has they have two different functions. The rough ER has a different function from the RER, or the RER has a different function from the SER. The rough ER has ribosomes, another organelle that's called ribosomes. Ribosomes are enzymes made mostly of RNA that make proteins. Is that clear? So these all along, this is studded with ribosomes that make proteins. This is a membrane that's continuous with the nuclear membrane, so they're connected. These are all connected to each other. From here all the way to here, they're all one giant membrane. And the endoplasmic reticulum has a lot of different functions, but obviously the RER, its main job is to prepare proteins, make proteins, and modify them. So the RER makes proteins and prepares them. The smooth ER, among many other functions, creates lipids. Let's not even say lipids. Creates vesicles to transport proteins. So, in order to get a protein from one place to another in the cell, you create these lipid bilayer spheres. They're made of, obviously, lipids. I hope it's obvious they're made of lipids. They're spheres. I can only draw circles. And in them, you will have proteins. Some of the proteins might be embedded in the membrane. They might be integral membrane proteins. Some of the proteins might be inside the vesicle. Some of the proteins might be actually on the inside. So these vesicles are moved all around the cell to transport the proteins where they need to go. That's how you get proteins from here to the membrane. Now, there's a lot of details that I'm leaving out. You'll learn them if you take AP Biology, but that's the basic idea. The RER is the place where a lot of the proteins are made. The SER packages them, helps them get ready to get sent to the next step in this process called the Golgi body. Golgi body. Body. B-O-D-Y. So there's a second. Hey, excuse me. Excuse me. Excuse me. Hello. Excuse me. Hello. So you have this Golgi body. Yeah. Shh. It's a Golgi body and it's spelled this way. Make sure you capitalize it. That's where these vesicles go. So they go from the endoplasmic reticulum to the Golgi body. That's the next step in their travels. All right. Have a good day. The cytoskeleton, and we walked through the video just a minute ago, and I'll post the video on Schoology as well to make sure you have it if you want to reference it. The cytoskeleton has at its center a centrosome. Is at the center. That center is next to the nucleus. We'll talk more about this when we talk about mitosis and meiosis, but it's a centrosome, and it's the center. It's the center of the cytoskeleton. Centrosome is the center of the cytoskeleton. Also, it has two. What? What were they? Centrosome and what were the other one? Centrioles. Right? So they have two centrioles. And they have a right and a left. Right/left. Right? So that's the cytoskeleton. All those are made, and the cytoskeleton is made of micro, that's the symbol for micro, tubules. I'm not sure I got the spelling right, but that's close enough. Now, that being said, we know that we have the rough endoplasmic reticulum and the smooth endoplasmic reticulum. That's where ribosomes on the rough, ribosomes make proteins if the protein has a lot, up arrow means a lot, of hydrophobic R groups, then it goes into the RER. And we should know that, and as you saw from the pictures, the RER is like these bunch of membranes that fold upon each other. There in the RER, they get things added to them. You add lipids and / carbohydrates to it's mostly carbohydrates, but whatever you add, you change the protein, and then it goes into the smooth ER. And there's further changes. Further changes. And then as the smooth ER gets changes happening to it, what happens is that you start at the end of it, at the end you have a bunch of Well, you have two kinds. You have the glycolipids. The smoothie R, I want to make sure you understand, is focuses on lipids. It does all kinds of fancy chemistry, but it's mainly about lipids, the smoothie R. Glycolipids and glycoproteins. They're going to be in a vesicle. This is another one of those vocab words, right? Vesicle, ribosomes, RER, SER. What's another one? Glycolipids, glycoproteins, centrioles, microtubules, centrosome, cytoskeleton. These are all words you should know, right? I could easily put on a quiz tomorrow. So when you're dealing with glycolipids, I put in vesicles. They're in vesicles. They're either in one of two places. They're either inside the vesicle, so they're getting secreted. That means they're leaving the cell. Proteins that are leaving the cell. Or they are proteins that are in the membrane.
I can't really see that part. It's the light.
The sun right there.
So we can't see.
Thank you.
So what you see is that you get this protein that's stuck in the membrane of the vesicle. These are going to be integral membrane proteins. When they get to the outer membrane, to the cell membrane, they're going to be what are they going to be? They're going to be receptors. They're going to be channels. Right? They're going to be what else? They're going to be binding proteins. They're going to be ID proteins, right? Cell recognition, communications, that kind of thing. What kinds of proteins are secreted? Well, those kinds of proteins, can you guess what's going to be put out of the cell? Can you make a guess? Any guess? I gave you one already in our discussion. You got mucus.
Oh, man.
So you put out mucus, snot, right? You got hormones. Hormones are
Sweat.
sweat,
Blood.
sweat, tears. Blood is not secreted from cells. Blood is made of cells and water and sugar and oxygen. So blood is a solution. Urine, urea, yes.
It's urea.
Urea is a salt that your cells release urea. Your kidneys concentrate it into urine.
Does saliva count?
Saliva. And there's stuff in saliva, but yeah, there's a lot of stuff in saliva, but saliva.
What about fingernails?
What is it? Fingernails, yeah. Yeah, the stuff, keratin. It's called keratin.
What about earwax?
Earwax, absolutely. Actually, earwax is a little harder, but let's go ahead and say earwax. But earwax is actually it's a combination of different things, but certainly some of it's being put out by cells.
Oh, eye boogers? I don't know what they call them.
Yeah, well, tears, the eye boogers are just the accumulation of dirt from overnight, and the tears get all dried up. But you can see that all this stuff gets put out by your cells, Signaling proteins, other signaling proteins. Have you heard of neurotransmitters? All right. your parents come in, I don't want there to be any kind of confusion as to why you're not getting it. Why is my daughter stressed? Your class is the only class that my child's stressed in. I'm just going to tell you straight up what I'm going to tell them. Number one, there are tutors available. They said the student response was, I just go home. And that's okay, but like I said, I can only do what I can do. That includes tutors, include high school peer tutors, include Case students, Case Western students,
What about you?
and myself. We're all here. Not today, though. Today I'm going to be leaving at 3:30, just so everybody's aware. I don't want any misunderstandings. Today there won't be anyone here after 3:30. I will be here till 3:30, though. So all that was available, and the response I got for doing an important assignment that you had 4 days to do, plus the weekend, was I just go home. And all the answers were available to you, all the explanations, and you chose not to do it, and the test is based on it. A good portion of the test is based on it. So I don't know how else to give you incentives to use the resources you have, but that's what we've gone on, and I'll make sure to communicate that as we move forward. So please try to take advantage of the opportunities that are given to you. Two, I really think you guys need to organize yourselves more and dedicate more time to biology We left off in this class, we left off after looking at organelles, the endoplasmic reticulum and the Golgi body. We looked at the pathway, which will be on the test. The pathway to secretory proteins, right? It's called the I can never pronounce it.
What's the title of this?
The cell.
Just say it?
The cell.
Oh.
I would wait to title something until it's done, until it's written. I would then read what I wrote, and if the title wasn't given, I would come up with a name that summarizes it all. That's how titles are generally written, by the way. So the secretory pathway is the idea that the proteins, these proteins we've been talking about and forever that we talked about, there were these amino acids that are all connected together in a line. They have these R groups with different kinds of R groups sticking off to the side. And remember, we call that the primary structure, which is the order of these amino acids. Amino acid 1, amino acid 2, amino acid 3, amino acid 4, that the orders of these amino acids are incredibly important for your understanding that every protein is different and that every protein has a different structure and a different function. Some proteins have to leave the cell. So when you have a nucleus and an endoplasmic reticulum and a Golgi body, and you have these vesicles leaving the cell that are full of hormones, or maybe you have hormone secretion. Maybe this is a cell in the pancreas, or it's a cell in a master hormone in an adrenal gland. It releases these things called hormones. Well, how did that? It's usually a protein. How did that hormone or steroid or whatever it is? How did it leave the cell? It has this membrane that we looked at in detail, and we looked at a 3-dimensional video of it. It has this membrane that it has to have a way of knowing this protein here that's purple is leaving the cell, but these proteins that are white stay in the cell. And these yellow proteins also stay in the cell. How do they know that? How does a cell choose? And it chooses right where it makes it. And we're going to that's one of our next units is protein synthesis. Well, maybe a couple units more. The first, remember that you make the protein here on the ribosomes. So the proteins are made here on the ribosomes. They're made here on these ribosomes in the endoplasmic reticulum. The endoplasmic reticulum is much more folded than that, but we'll go ahead and just pretend like there's a lot of folds here. You saw the endoplasmic reticulum when we looked at it in 3 dimensions. I posted the video on it in Schoology, so you have access to that. So you have these ribosomes. These ribosomes, they start to make those amino acids. And as I said, if you have a large group of hydrophobic amino acids being made right away, they start to follow the secretory pathway. They go from here through the endoplasmic reticulum, through the rough endoplasmic reticulum, then through the smooth endoplasmic reticulum, where they're changed and things are added to them. And then they get pinched off. The vesicle gets pinched off. So you get this vesicle, and inside the vesicle are proteins that are leaving. They're leaving the cell, and inside the vesicle and in the membrane of the vesicle are proteins that are staying in the membrane when it gets there. There are these membrane proteins that we talked about. They could be all kinds. They could be receptor proteins. They could be anchoring proteins. They could be proteins that are enzymatic. They have some enzymatic function. They could be proteins that are channel proteins. These vesicles. Oh, ungroup. Okay. Oh, that didn't work. Undo. Forget it. I'll just draw it again. These vesicles move, and they get walked. You saw the little walking proteins that move these vesicles from one place to the other. And these walking proteins get walked, not right to the membrane from the SER, but to the Golgi body. So I'm going to go ahead and erase from here and move to the other side. So just for...
Can you repeat that again?
So the vesicles get walked, not directly to the membrane, but to the Golgi body. And the Golgi body is another organelle that's where this vesicle is going to fuse to it, and these proteins are going to enter a whole nother set of changes. There's all kinds of changes that happen. We don't even know all the changes that happen at every step along the way. But this is a pathway, a road that these proteins, where they start here, and they finish out here. And at each time the vesicle fuses, like two bubbles coming together in air, right? They fuse, proteins are changed in the membranes, they travel through the membrane, inside the endoplasmic reticulum, inside the Golgi body. And then when they leave the Golgi body again, another bubble or vesicle comes out, and you have the proteins are in the vesicle, and they're being walked to the membrane.
The Golgi body fuses or what?
What does the Golgi body do?
What it fuses.
The vesicle fuses, you see. The vesicle gets walked along the microtubules right to the Golgi body. And then when it gets to the Golgi body, the membrane of the Golgi body and the membrane of the vesicle, they fuse. Like two bubbles coming together. I showed you pictures of it. Please review the video I posted on Schoology. Watch the vesicles fuse. Watch the proteins get transferred. Say what? I'll post this as well, yes. The whole thing, including our discussion. All right. And after it pinches off, it goes, let me zoom in here. When it reaches this cell here, when it reaches the membrane, this vesicle fuses. Now I'm going to go ahead and take a close-up look at what happens when it fuses. Let's take a look at it here. And what happens is that you have a membrane. I have to do it in a time. So there's the membrane. Here's the vesicle. I'm just going to draw a circle if you don't mind. You have to imagine. I don't want to do this, but I'll go ahead and draw little circles. It's a membrane, right? So this is all a ball of lipids. It's correct? And here's this other membrane over here, the cell membrane, that's, again, a ball of lipids. But the ball is much bigger, right? So it's all around the cell, and they're phospholipids, so we know that they have these hydrophobic tails that stick in. We know the hydrophobic tails are in the ball of lipids, too, in the vesicle. That's what we call a vesicle. Is that clear? You drew this for a reason. I had you do this on purpose. It wasn't an accident. Now, this membrane is going to come into contact with this membrane, and they're going to fuse like two bubbles. And these little proteins that are in here, in the membrane, they're proteins. They have different functions. Maybe one's a channel protein. Maybe one's a signal protein. Maybe one's a receptor. And then, of course, on the inside, you have the proteins that are going to be kicked out, right? So when we move a little further, give it some time. And what happens is the vesicles start to fuse. So oh, jeez. Well, all right. Watch the video that I posted on Schoology. This is, I'll try to get this part. So just to make sure you understand here, what happens is the membrane fuses what was inside the vesicle. This is a cutout of this, right? What was inside the vesicle goes out. What was in the membrane of the vesicle now is part of the membrane of the cell. That's how you get proteins from inside the cell into the cell membrane. Yes?
Those are hormones? Like they're
These could be hormones, yes, or they could be signaling proteins, or they could be waste. It could be urea. Could be let out of here this way.
Why do you have hormones right there?
Because this was just an example. Yeah, these are hormones.
Okay.
But I'm just saying, just don't stick with the idea it's always hormones.
Okay. So it can't be.
It can be hormones, yes. Restroom? Yeah, go. This is all recorded. I'll post in just a minute after you leave. After you leave, I am covering. Does anybody have Mr. Custer's class, 4th period? No, right?
That's but not fair.
Yeah, I know, but sometimes some 10th graders have them. But if you had them, you wouldn't have me. But I just want to make sure because I'm covering his 4th period class. All right, so that's the pathway you have to know. Right? Yes. I'll post it all as one. I want to edit it first. Now, so far we've talked about the cells. Again, I'm trying not to say the stuff that I've already said just because. Yeah.
This is for the Halloween contest.
Waiting for some reason. So if this is the cell membrane here, then again I'm going to go ahead and do a time lapse, and I'm going to. And so when we look at the cell, as the stuff is going in, if you have this thing that's trying to go in, it's too big to fit through the channels, then the membrane will literally bend. Then what you'll get is the membrane will start to invaginate. That's a word that we use for bending or folding. And it will start to make a pocket. Now you saw what I'm going to expect is you're going to be able to draw this kind of eventually. So this is after some time, the membrane will start to fold on itself and create this place where the thing can be, and we saw it happen with and this is time, of course. And we saw it happen with the amoeba, as the amoeba folded its membrane and absorbed the little creatures, the bacteria. And what will happen is eventually and this is again part of the membrane. I'm drawing it quickly, I'm sorry for the mess. And I'll fill in the lipids in a minute. But more time. There'll be a time when the membrane, just like the endoplasmic reticulum, in the opposite direction, just like the other organelles in the opposite direction, when things are leaving, they're going to form a vesicle, and in that vesicle is going to be that protein. That vesicle pinched off. It pinched off of the main membrane, the cell membrane, and inside is the protein. So we have this membrane that's pinching off. If you put the grades in the grade book, and then we can pass them back. the membrane, then they get this vesicle, what we call a food vacuole, because this was food, or maybe it was a signal that was sent by another cell, whatever it is. It's going to be, the membrane's going to stay intact. So you see the membrane was straight, and continues to be straight. But inside now you have this this vesicle. You know, this, we call it a food vacuole. And a food vacuole is just one of these vesicles that's going to get transported. I'm putting it on there. It's a vesicle. We talked about them, right? That's how we got stuff out of the cell. And this is going into the cell. So this is inside. And this is outside over here. And over time, the food, I guess I should reverse this. Thanks. So what you see is on the outside, you have the food that needs to get in the cell, and this happens every time you eat something. The membrane bends, folds, surrounds the food, and so that when you have, or anything that comes into the cell that's too big, then what you have is this vesicle, right, that gets transported, it gets moved, it has little walking proteins that walk it where it needs to go. You saw them, I showed them to you. And inside the vesicle is food. Well, that's great, but how do you digest it? How do you get this food that's in this vacuole digested into all the parts of the cell that need to eat it? Yeah, you're good. Enzymes is right, but they can't just be they have to get inside, right? Because this is the cytoplasm, right? All right, we're done with the quiz, guys. You have to turn it in. Thank you. So, this is the vesicle. It's inside the cell. Now, you have to remember, we're talking inside the cell. So, the nucleus is over here somewhere. And here you have the endoplasmic reticulum. There's the rough, right, with the dots. And here's the Golgi body over here. And then so here you have this vesicle. Well, now everyone's done the quiz, right? So the pathway that I would have seen you draw that would have got you 100% on this question would have been start here, proteins start here, and they follow their way through the endoplasmic reticulum. They go to the Golgi, then from the Golgi they go out. They fuse it, vesicles fuse at each time, and this is end, right? This is the end. So they start in the endoplasmic reticulum, end outside. It's just that easy. That would have got you that question right. Now the vesicle's here. How do we get the enzymes inside? You're right, we need enzymes to break them down. They need to hydrolyze them. How do we get the enzyme? We don't want enzymes that are going to break food. We don't want enzymes that break up amino acids and lipids and DNA in here, right? Why don't we want those enzymes just floating around in here? Because it breaks down yourself, right? Your cells start breaking down. That's what happens when you die. These enzymes get released, and you start. Why is anyone on a screen? Everyone should be looking up here and being part of this discussion.
Can you zoom in on that last part?
Yeah. So what here?
No, over with it, yeah.
All right. So that's all in the previous video. It should be all in your notes already. The way through the secretory pathway. But when we're looking at, and let me tell you this, those of you that take pictures of the screen, again, I'll post the video, but the problem is taking a picture and then trying to memorize it from a picture is not as useful as drawing it. So as I'm sitting here drawing it, you should be drawing it too. It would make your life a lot easier and you would learn more. But I can't force you to do anything, so that's my suggestion. So we have here this, how do we get the enzymes in the food vacuole? This is called a food vacuole that the cell just brought into itself. How do we get the enzymes in this food vacuole. Well, the proteins had to start here because that's where they made these enzymes. We don't want them out here, so we keep these enzymes inside the endoplasmic reticulum. They're up this smooth. They go to the Golgi body, just like the ones that were secreted are going out. But these enzymes aren't going to get secreted. These enzymes are going to go from here, from the start, through the endoplasmic reticulum. They're going to go across. They're going to go into the Golgi body. But this time, they're going to come out this way. And they're going to pinch off and they're going to make vesicles just like they did to leave the cell. But this vesicle is going to be a very special vesicle with really, really strong, very powerful enzymes that break down just about everything. They break anything that they come into contact with down. And these enzymes sit inside here. And this is called a lysosome. This is an organelle called a lysosome. And this is full of digestive enzymes. It came from the Golgi body. The proteins, the enzymes were made here in the endoplasmic reticulum, the rough endoplasmic reticulum, by ribosomes. They traveled through the endoplasmic reticulum pathway. They went to the Golgi body. They pinched off, just like the ones that were leaving did. But the Golgi body labeled these differently. They labeled the membrane differently so that the vesicle did not leave the cell, but rather just floats around and waits for a food vacuole. When it meets up with the food vacuole, they fuse. So you have one common vessel. Now they have one big vesicle with food and lysosome because they fuse together. And once that happens, what do you think happens to that food? It gets broken down. That's right. And so you have this giant vesicle here that had the lysozymes, the enzymes that were in the lysosome, and then they have the food, and that food gets broken down. And that's how you digest. That's the end product to digest. What comes out of this thing is, once it's done, what comes out of this thing is amino acids. What comes out of this thing is carbohydrates, sugars, right? Simple sugars, monosaccharides. Right? What comes out of this thing is nucleic acids. That's what you do with the food you eat. You make it, you ever heard that thing, you are what you eat? Yeah. Well, that's what we're talking about. You're talking about having nucleic acids but you're talking about the monomers now. If they fuse with a lysosome, they're no longer polymers. They get broken down. So you're talking about nucleic acid monomers like A, T, G, C, U, right? Any of these. You're talking about simple fats, so fatty acids. So you're not talking about triglycerides at this point. You're talking about simple fatty acids. Either they're saturated or they're unsaturated. Either way, single or double bonds. But that's all that comes out of those food bags. So every time you eat, you take that burger, you break it down. You completely tear it apart. If there's a problem with your quiz, do not bring it up with your TAs. Bring it up to me. We will discuss it. I will look at it. I'll tell you if it was a fair grade or not. I can tell you these girls know what they're doing. I gave them the key. They're following my directions. But if there is a mistake, we're all human. We all make mistakes. Come and see me. All right? All right, are we all good? Does this make sense to you? What? Fatty acids?
Yeah.
That's fatty acids.
Oh.
Can you repeat what comes out of the...
Sure, I think that's a fair thing to say. So what we have here is 1, amino acids come out of the food. 2, monosaccharides come out of the food. 3, nucleic acids come out of food. And 4, fatty acids come out of food. So lipids, and I shouldn't even say fatty acids because it's cholesterol too. I should call them just lipids. Simple lipids, right? Broken down lipids. Let's just say lipids. I think that's a better answer. And that's what you eat. That's why you eat, to get these things. Is that clear?
Lipids and levels.
Lipids and all the different kinds of lipids. So cholesterol, fatty acids, that kind of thing all come out of food. And we also, nucleic acids, right? And monosaccharides and amino acids. We've broken down the polymers. Yeah.
I can't read what that says.
Which one?
2.
2 says monosaccharides. They're monosaccharides. Let me print that clearly.
I just needed you to zoom in more.
Oh, okay. I can print it bigger though, I think.
I can't see.
Well, I'm in the same boat. My eyes are old. All right, so are we good? Does this make sense? So we talked about things coming in. By the way, there's a term for things coming in. There's 2 pathways here. There's exocytosis, which is what? Exo means out, very good. And then you have endocytosis. That's right. Food is not the only thing that goes into a cell, but food is a big one. So we do have to get food into a cell. And so this is how we transport things. Just like we transport things in the real world, out here in the big world, we transport things in containers, right? In shipping crates and stuff. Well, in your cell does it in these vesicles. Whether they're going into the cell or they're leaving the cell, it doesn't matter. It's always a vesicle pinching, being transported, pinching off, being transported, and fusing, mixing, out comes the product, that kind of thing. All right? This is the process of bringing things into the cell and out of the cell when they're big. On Monday, we'll go back and cover again osmosis and diffusion and make sure we understand that because you're going to have some problems on that. I'm debating, I'm going to talk with my AP kids and see if it makes sense to do the water potential equation. Sometimes I think I should cover it with you guys just because it's honors and it would help you get ready for AP later. But part of me is like, it's a lot. So anyways, yeah, you have to actually try though before we get it. All right. Don't worry, I'll cut that out of the video. Recorded that this endocytosis, this special type of endocytosis is called phagocytosis. It's a special type of endocytosis that allows us to take big things into the cell and then digest them, break them down, or whatever we're going to do with them. In this case, it's a food vacuole. Sometimes it's a signal molecule, but we'll talk about that in AP if you take AP course.